Okay, so this is the floating leaf disc assay, also known as the FLDA assay, or FLDA Lab, for investigating the effect of light intensity on photosynthesis. The materials for this are pretty easy. You need some sodium bicarbonate, which is baking soda, a balance, of course, because you're going to be making a solution, weigh dish and spatula to make the solution, some liquid soap, a plastic syringe that's 10 cc's or larger, distilled water. You need to have a chloroplast source, so we are using spinach leaves, and a hole puncher in order to turn those leaves into discs, stir rod, pipette, beaker, plastic cups, a ruler or a meter stick, because we are going to be testing the effect of light intensity by varying the distance of these chloroplasts, aka spinach leaf discs, floating leaf discs, from the light source. And we need a light, so we're using flood lamps. And this lab also includes a second part where lab students groups investigate an additional variable other than light intensity. For all intents and purposes, this demonstration is just going to show the straightforward light intensity as the variable. So if we look over here, what we have in these cups, we have some spinach leaves that have been cut out using a hole puncher. They're in uniform discs. Each cup is in a solution of sodium bicarbonate with some dish detergent, measured to about 300 mils as the lab calls for. 10 discs in every cup. Every cup has been controlled, with the exception of the control. I'll show you that one in a minute. This is not a control. This is a test cup. The control does not have any sodium bicarbonate that has been infiltrated into the discs. The cup is set up the same, but the discs have been treated differently. These discs have been vacuum infiltrated with a carbon source using the baking soda, the sodium bicarbonate. That's what the 10 cc syringe is for. And after we've infiltrated the discs to make them carbon heavy so that they sink, as we can see here, we put them into these 300 ml sodium bicarbonate and soap solution cups, and we are setting them underneath light sources, flood lamps that are set at different size, at different heights. So here, this is the test for 50 centimeters. We've started a timer about 5 minutes ago. The lab runs for up to 14 minutes, and it calls for us to start counting and measuring the time at which discs start to raise, which indicates rate of photosynthesis. So if you're looking at the rim right here, we've got that cup right there set at 50 centimeters going down the lab bench. That one is set with a more intense distance, so that's 100 centimeters. And then almost from ceiling to floor with that cup down there, that's 200 centimeters. These have been measured, and again, we are watching a clock, and we are going to be recording the time at which these chloroplasts, or these spinach leaf discs, start to raise. So here we already have, this one is at 30 centimeters. And we have 1, 2, 3, 4. This is at about minute, probably 5. And the lab asks us to stop once we have half of the discs in each cup that have floated. So at minute 5, we almost have the 30 centimeter completed. This is the 3 centimeter. And the 3 centimeter has 1 that is raised, 2, 3 that are on their way up. We're gonna call this soon. We're gonna just take this out. This is very close to the light source, as we can see. And we have many that have raised here. Once that's at 5, we've got number 4 coming up the side right here. We'll stop that and record the time. And over here, we are running a control. So the control is set at the most intense light, which is 3 centimeters. But these discs have not been infiltrated with a carbon source. So there's no carbon that's been introduced to these chloroplasts. So we will see what happens with these ones. So what we're going to do for the rest of the lab is walk around and just kind of keep time and mark the time that we have at least 5 discs in the cup that have raised. That's 50% photosynthesizing, and that is our benchmark. Okay, so that last bit was the overview and setup of how the lab looks as it's performed in our AP Bio classroom. As a full class, the lab is easily divided up into a team effort. Each group concentrates on just one distance from the discs to the light. But this is such a simple experiment using what we refer to as just kitchen chemistry, techniques and materials, that the entire thing can easily be performed at home for a student that maybe was absent. And that's what I'm showing you here. For a home investigation of the effect of light intensity on photosynthesis, you need basically the same materials, more or less. There are some substitutes, and I'll show you here. You need a single hole punch. You need to have some green leaves. I'm still using spinach as the class uses. You could use any green leaves. You could go out in your yard, your neighborhood, go for a walk, just find some green leaves. Baking soda, we're using baking soda still here at, in our at-home demonstration. That's our carbon source. You need to have water. So I've got some water in a pitcher right here, and I'll tell you about how we're going to expand the volume so that way you could do all of the cups at home instead of just one. It really should be distilled water. Distilled water can be bought at the grocery store. It's $1 for a gallon. I'm doing this on the fly. I just decided that I was gonna do this morning. So I'm using regular tap water, and that would be okay too. Just realize that, you know, the results might be slightly changed, although the overall trend should stay the same. You need to have a syringe. So in the lab, we have tons of these. You might have one of these kicking around your house. You know, it's the same medicine syringe that you get from the pharmacy if you ever have a liquid medicine or prescription. So you probably have some of those maybe in a medicine cabinet or a junk drawer. So you need to have a syringe. You need to have some kind of light source. I'm just using a desk lamp, so some kind of portable, lightweight lamp because you're going to be moving this to different distances. Dish soap, so just some liquid dish soap, a cup, so I've got a cup right here, and you need to have you know, something to measure out the baking soda. So I'm just going to use a teaspoon. I know in the lab it says 1 gram for one of these setups. And again, we're going to modify that so that way you're doing all 6 at home. But, you know, at home you probably don't have a balance. I, of course, have a balance at home. So I'm going to show you what that mass looks like for the baking soda on a teaspoon. So that way you can just try and reproduce that amount at home. You also want to have a measuring tape, so that way once this is all set up, you can go ahead and start varying the distances for the light. So you want to get a measuring tape as well. Okay. As the lab at school would have been performed, one lab group would have been doing one of these distances. So you only had to set up 1 cup, which equals 10 of these discs. Of course, at home, you have to do the whole lab on your own. Oh, it's very easy and it's still really quick. It's just you have to multiply everything by 6 because you have to do all 6 of the different setups. So just grab your leaf and your hole punch and start punching away. It's really quick. 4, 5, 6. Keep going until you have 10. You need 60. We have way more than 60 in here. So that's how you're going to get your discs. Okay, so the next thing is we need to measure out our carbon source. We're using the baking soda. Again, I'm sure you don't have a scale at home, but you might have actually a kitchen food scale, and those actually measure the grams too. So you needed 1 gram of baking soda to 200 milliliters to do 1 setup. So we need to actually multiply that all by 6. So instead of 200 milliliters of water, you're going to need 1,200 milliliters of water. And instead of 1 gram of baking soda, you're going to need 6. So if you have a kitchen food scale, great. You can measure it precisely. If not, since it's kitchen chemistry, it's give or take. It's just an estimate. So I'm going to go ahead and use my scale at home, just show you about how much that would be on a tool you probably do have in your kitchen, which is a teaspoon. So I'm just using, I don't have weigh paper here in my kitchen, but I do have index cards. So I'm going to zero that out. Got a 0 on my scale, and I'm going to just keep pouring this until I have 6. As you can see, it's really just a little bit, so I'm going to take some of this off. We have just about 6. Good enough. So 5.9, we're gonna call that 6. So that's what 6 grams looks like. So if we were using a teaspoon, I'm gonna say that's probably close to a full teaspoon. All right, so I mean, you're looking at a level teaspoon to do this at home. That would be 6 grams of your sodium bicarbonate. So the next thing is we want to make this solution. So again, at school for 1 cup, the directions said you needed 200 milliliters of distilled water. And again, I'm just using regular tap water, to 1 gram of the sodium bicarbonate and 1 drop of dish soap. And we are, of course, making that 6-fold. So instead, this is 1,200 milliliters of water, which is in just a little pitcher. I did use a measuring cup, and the equivalent, and you could Google this, that's exactly what I did, but 1,200 milliliters is equivalent to 5 cups. So you need 5 cups of water. So that's been measured out in here. So we're gonna add this into a bowl. Room temperature water is better, right? Because that's not one of the variables. We're not doing cold, we're not doing hot. So this is like warm, room temperature. We're gonna add the 6 grams of baking soda. And then I'm gonna add 6 drops. And at school, we would measure this very precisely with a pipette, but precision's not really necessary. So that was 1, 2, 3, 4, 5, 6, and 7. It's fine. Take your teaspoon, give it a mix, and that's our solution. Okay, so now it's time to go ahead and get this all set up. So it's always a good thing, you know, you watched me just stir it, but it's always a good thing to go ahead and stir it again. Make sure your solution is nice and stirred. So you're gonna take that syringe that you had, and you're gonna take the back out, that's called the plunger, and you're gonna take your collection of these little uniform discs, and they might be clumped together, and so I'm just counting them. That's 1, 2, 3, 4. You're gonna put them right down into the syringe. That's 4. That's actually 2. So there's 6, 7, 8, 9, and 10. And sometimes when they go in, they stick to the side, so you might have to give it a little bit of a tap, and that's okay. Just make sure that they do all make it down at least. You see how I have one stuck right there? That's fine. Just don't put the plunger down to crush it. So that's probably about how far I'm going to bring it, because I can't really get that one down. And then you're going to come over to your solution, and you're going to draw up a little bit of liquid, just a little. You want to still have volume left to make the vacuum. And at this point, anything that was stuck, see how I just swirled it? Anything that was stuck in there is going to go ahead and come off the wall. Turn it upside down, and you still have volume to go ahead and pull. We're going to make a vacuum, and so you're going to put your finger over the top, and then you're going to pull. You're not going to pull the plunger out, but you're going to pull it down, and then you're going to let that stay for about 10 seconds. You're going to let that sodium bicarbonate kind of do its job, and I want to make sure that I've got as many of those discs, there we go, into the liquid. And you can pull it back, don't pull it out. The longer that you hold it here, the better you're infiltrating all those empty air spaces with sodium bicarbonate. It's probably been about 10 seconds. I'm gonna keep holding the plunger, but I'm gonna release my finger. You'll hear like a little puff of air. And then you can give it a swirl. And what you're trying to do is get these discs to float to the bottom. It doesn't always work on the first time, so you might have to do it again. Do it again, just bring it up, put your finger over it, pull it back, make sure that it's all in there, count to 10, and then you'll release it and you'll swirl it, and you'll keep going until these discs fall down to the plunger. You're trying to get these to sink. So this has been 3 tries of me trying to infiltrate. And as the lab says, 3 tries is usually okay. It very rarely will you get all 10 discs to sink and infiltrate on one vacuum. 3 tries though, and they really should go down. And you see I really am getting a lot of them to come down. If they're not sinking, you probably need a little bit more soap. You know, and dish detergents vary. Some are more viscous than others, and here you go. Sometimes it's just being patient. So these are actually starting to fall. If you're finding at home that they're not falling, even after 3 tries, or one is stuck up like that, go ahead and go back to your solution and add another drop or 2 of dish detergent, and go ahead and give that a swirl. And then you could go back and start again. But here's that last little guy. So he's going down, and this was 3 tries. You just had to be patient a little bit. So this is what you want it to look like. And again, if you were in school and you were running this lab with your lab partners, you would only be setting this up once. This would be done at this point. You'd be going ahead and adding these into the cups, which I'm going to show you next. Of course, at home, you are multiplying this, and you are going to do this exact same procedure times 6, because you have to do 6 distances all on your own. But that's what this syringe should look like. So this is 10 uniform discs, all infiltrated. Is we're going to set up one of our cups. And so you're going back to, this is why we had to make so much. It would be silly to have to make so much just for 6 little syringes, but you need to be able to make 6 cupfuls. And it really is important that each time when you're done, you do go and, you know, toss any of the solution that's in this cup into the sink and get new, because you don't want to be reusing the same solution after it's already been sitting in the light. So you always want to go back, make sure you're controlling that variable that you're taking from a stock solution all 6 times. I guess in the best case scenario, if you had 6 different little lamps and you could just set it all up and run it straight through, you could do it that way. I don't, so I'll be doing this 6 separate times. So the lab says that you would be filling up a beaker, a 250 ml beaker at school. This is about a 250 ml beaker. It says 3 centimeters from the bottom at least. That's not really what's important. What's important, again, if we're controlling variables, is that we are making sure that we are always filling these to the exact same depth. So here we go. I just want to make sure. And I'm going to make sure that every single time that I run this, I'm going to put about 2.5 inches, or let's just call it 6 centimeters. So I know it said you needed at least 3, so I'm going to do 6. Oh no, that might have been a little bit more. Yeah, 6. So I'm going to make sure that I always have 6 centimeters. So there's my cup that's got the dish detergent, the water, and the sodium bicarbonate. Now what I'm going to do is I am going to take this plunger out, and I am going to release these discs into that solution. Make sure all 10 went in, and if you come down here and you look, they're all going to sink. So in here I have those discs, and they're all on the bottom. This is what you want your setup to look like. From here, I'm gonna go ahead and put it under light. Okay, so picking up where we left off, this is my cup. It has 6 centimeters or so of this solution. It has my 10 discs. They're all sunk to the bottom. I have my little desk lamp, and there are a total of 6 runs that we do with this. And so we're testing 5 distances plus a control. So the first distance is very intense, and this is going to be 3 centimeters from the light source. Then we're also going to do 30 centimeters, 50 centimeters, 100 centimeters, and 200 centimeters. Okay, so going back, and I just want to make one comment about the control. The control is actually going to look just like this setup. So it's at the most intense light, it's going to be at 3 centimeters. The difference is your 6 cup, which is going to be your control, is not going to have any of the carbon source. No sodium bicarbonate when you prepared that dishwater soap solution. It's going to be just the water, just the dish soap, no sodium bicarbonate. You follow the same procedure. You set it up at 3 centimeters, and that's your control. Okay, so this is where my tape measure comes in, and you want to measure from the top of the cup to where the lamp is. This is 3 centimeters, and you also want to have a record of time at this point, because what you're doing in this lab is you are going to count how many discs are raised, how many have gone ahead and floated. That's why it's called the floating leaf disc assay. These discs, as they photosynthesize, are going to raise to the top, and every minute you're going to stop and you're going to take a recording as to how many discs are raised. And you're going to keep doing that until 50% of the discs have in fact floated, so that would be 5. For any of these cups, as soon as you have 5 discs, you can stop your time, but every minute you do want to record. So as soon as you turn the light on, you're going to start your timer, and also it should just be noted, you know, you want to do this in the absence of other light. So I've gone ahead and shut off all of my other house lights, so that the only light, of course, we've got light from the flash of the camera, but that won't be going on throughout the entire experiment, is going to come from this lamp. So I'm going to go ahead and turn this on, and this is the most intense 3 centimeters, starting at about 11:49. So after 1 minute, none of the discs have raised. So it's hard to tell with the bubbles, but at 2 minutes, we do have 1 disc. One just floated right to the top. I'm gonna just see if we can see that. If I just move that out of the way for just a second, there you go. So you can see you've got 1 at the top. And that was at 2 minutes. So now we're gonna watch, because once 1 starts to raise, they usually kind of go fast. So that was at 2 minutes. And there's another one lifting in the back. There we go. So that one's raising now. And, you know, for this one, this goes fast, right? Because this is the most intense light. You do really want to watch it very carefully. There's 1 raising. For the other ones, as you're going to see, is, you know, time just ticks by. And it's like watching water boil. So what can you be doing while you're waiting for these little discs to float? If you have more of the same type of cup, like I do right here, you could go ahead and start preparing, because, you know, you have to do them for 30 centimeters, 50 centimeters, 100, 200, plus a control. And you can go ahead and you can use that syringe, and you can start infiltrating more solution with the discs and getting those to sink. So you can be prepping the other setups while you're waiting. So at about 2.5 minutes, we have 2 discs. So just about coming up on minute 3, and we have the 3rd disc that's starting to float. It's almost at the top, and that's minute 3. So 3 discs at minute 3. It's almost been a disc per minute. 1 disc around minute 1, the second one just after minute 2, and that 3rd one after minute 3. And this 4th one is very slow to raise. It's been 5 minutes. There it goes, very slowly, but it's gonna get there. And so here we are, it's been 8 minutes, and that is going to be our 5th disc to float. There's a 6th one lifting on the bottom, but at this point, that's 50% done. This is the stopping point. 5 discs floating, 8 minutes for 3 centimeters. That's considered the most intense setup. So you will now repeat and follow the same procedure, keeping time in a data table for the other 5 cups, 4 more tests, and then 1 control. Make sure that you control all of the variables so that you're using the same amount of solution, using the same lamp, and you know, make good use of time in between, because clearly if it took 8 minutes for these discs to raise in the most intense light, you can tell that this is just a tedious. It's not a difficult, but it's a time-consuming lab. It's why in school we do break it up into 6 lab groups, and you know, it's a little bit of divide and conquer and a team effort, but it's certainly something that you can easily do at home. If for any reason you don't have any of the materials, come see me. I have hole punchers, I have syringes, I even have baking soda that I could lend you out. So this is definitely something that if you are absent, you could follow this demonstration and go ahead and do at home. A final component that you have to do, something that is an experimental design. And so You know, you have to test something other than light intensity. So that's part 2 of the lab. I'm not going to do any of that for all intents and purposes. This was a demonstration just for the straightforward light intensity portion of the lab. Some good ideas that you could easily test at home would be maybe to go outside and pick different colored leaves. It's fall time when we do photosynthesis in AP biology, so maybe grab some colored fall foliage leaves and then set up the exact same experiment and see if color of the leaf changes. Maybe Google a different carbon source that you have readily available at home other than baking soda. Maybe alter the soap amount. There's lots of different things that you can try and do. Maybe you can get your hands on a CAM plant or a C4 plant. Maybe you can change the different color of light bulbs. Maybe you have access to a black light or a red light or a blue light, or maybe you can try different wattages, you know, a 40 watt bulb versus a 60 watt bulb versus a 100 watt bulb. So there's lots of things that you can vary for a second experiment in addition to doing light intensity. So have fun, good luck, don't forget to take good data, and if you have any questions or you need any materials, be sure to see me.